Welcome to the review on mutations and nature of a gene. Problem 1. A wild-type male fruit fly was exposed to UV radiation and subsequently mated with an unirradiated female. One of the female's X chromosomes carried a dominant mutation for the ebony body color trait and several inversions, which prevent crossing over during meiosis. Now, I highlighted that fact because that means that we can ignore any possibility of crossing over during this whole problem. Then it goes on to say that many of the F1 females from this cross had the ebony, multiply inverted chromosome from their mother and an irradiated chromosome from their father. So let's stop there and just diagram this cross. So we're talking about the sex chromosomes, right? We're talking about X and Y in fruit flies. So let's draw the parental generation first. So we know that the male was exposed to UV radiation, so we're going to draw his X chromosome as this squiggly line to show irradiation, and his Y chromosome is drawn like this. And he was crossed or mated with an unirradiated female, so she has two normal X chromosomes. And it says one of her X chromosomes carried a dominant mutation for ebony body color. So we could draw that as an E. When they're mated, it says that many of the F1 females from this cross had the ebony multiply inverted chromosome from their mother and the irradiated chromosome for their father. So let's just focus on these females. So they had the irradiated chromosome and the chromosome with the dominant trait for ebony body color. Now it says when these females are crossed with normal males, most of them produced ebony and wild-type sons in equal proportions, which means that, as you know, they only give one X to each son, so half of the sons got the X chromosome with the ebony body color trait on it, and the other half got the irradiated X, which gave a wild-type. And then it says 2 F1 females were exceptions to the norm. Female 1 was crossed with a normal male and produced as many sons as daughters, but half of the sons had ebony bodies and the other half had veiny bodies. So now we're looking at 2 F1 females. So I'm going to write it as F1-1 and F1-2. Now remember, these were crossed with normal males. So it's saying that the first F1 female, when crossed with a normal male, had as many sons as daughters, so we can write a ratio of 1 son to 1 daughter. And half of the males had ebony bodies. And the other half had veiny bodies. Now, the F1-2, the second female, when crossed with a normal male, produced twice as many daughters as sons. So we can write 1 son to 2 daughters. And all of the sons had ebony bodies. So for the sons, 100% were ebony. Now it wants us to explain these results. So what you might be thinking is, obviously these things had to come from mutations, right? Which, after being exposed to UV radiation, that's definitely a mutagen. So that can explain all of these different traits that are coming up. Now you might think if a male fruit fly is exposed to UV radiation, then all of his progeny should have the same mutations. However, different cells and the DNA in different gametes, the sex cells here, could have received, they all received the same radiation, but they could have been mutated differently. So we know that most of the gametes didn't have any really effect with the UV radiation. And that's because if you see in the F1 generation, they all had this irradiated chromosome, but then in the F2, most of those females didn't pass on any mutations to their offspring. However, these 2 are exceptions, and they're the ones that came from gametes that actually were mutated. So let's look at this F1 female. Now remember that these are the F2. So the mutations from the parental generation from that father, the irradiated X chromosome, didn't actually show up until the F2 generation when you're looking at the males. And remember, that's because males in flies are hemizygous for all traits on the X chromosome because they only have 1 X chromosome and 1 Y. So it's saying that half have ebony bodies, which comes from that original unirradiated X chromosome from the parental mother, and the other half have veiny bodies. So to explain that, we must say that this F1-1 female had a mutation for veiny bodies on her irradiated X chromosome. So we're going to draw that as a squiggly. Now, was this dominant or recessive mutation? Well, if you see that the half of the sons have the ebony bodies, so if the veiny bodies was dominant, that means that that mother would exhibit this veiny body, but we're saying that all of the F1 females that we're considering only have ebony bodies. So this is a recessive mutation, and that's why it only shows up in the hemizygous male of the F2 generation. Now for the F1-2 female, it's saying that twice as many daughters are seen as sons, which means that half of the sons were dead. So that means that she must have had a mutation that was a lethal mutation. So mutation for lethality on her irradiated X chromosome. And again, this was also recessive, because if it was dominant, then that F1-2 female wouldn't even be alive. So we know that both of these mutations are recessive, and they're both on the irradiated chromosome, and they came from the UV radiation treatment. Problem 2. Flower color in a certain species of plant with a diploid number of 36 is determined by a pathway involving 4 enzymes. The gene for each enzyme is located on a different chromosome. The figure below shows that either of 2 different enzymes is sufficient to convert a yellow pigment into an orange pigment. A pure-breeding pink-flowered plant is crossed with a pure-breeding yellow-flowered plant. All of the F1 offspring have orange flowers. Determine the phenotypic ratios for the F2 offspring if F1 plants are allowed to self-fertilize, assuming the pink-flowered plant is mutant in only one step of the pathway. Show the genotypes for P and F1 plants, indicating which genes correspond to each biochemical step. So let's look at this biochemical pathway. We have a white pigment going to a pink pigment, which then goes to yellow, and then either of 2 different enzymes can convert yellow into orange. So let's write the gene products. We can pick whatever letters you want, and these will also be the alleles that we're going to write in the genotypes. So from white to pink, we can call this gene product, let's say W for white. From pink to yellow, we can call that P for pink. And then from yellow to orange, there are 2 different gene products here, right? Because either of the 2 enzymes can do this conversion. So we'll call one Y, let's say, for yellow, and E is the next letter, so we can call the other one E. Now let's look at the specific things they tell us. So they tell us the diploid number here. So here we know that this plant is diploid and is therefore going to have 2 copies of every allele. Right, and that's normal when we do human genotypes, and everything that's a diploid organism, they have 2 of every chromosome and therefore 2 copies of every allele. And then they also say that the gene for each enzyme is located on a different chromosome, so all of these genes are unlinked, so that's good. Simplifies things a bit. Now let's look at the cross they did. They did a pure breeding pink flowered plant and a pure breeding yellow flowered plant. Now what could be these genotypes? Well, remember that for pure breeding plants, they're homozygous, either recessive or dominant, at every allele in their genotype that we're considering, or else they wouldn't be pure breeding, right? So for a pink flowered plant, that means they are stuck at this pigmentation in the biochemical pathway, which means that they must be lacking this gene product W. They must be lacking gene product P, because they're stuck at the pink step. They can't convert it to yellow. So let's write that. They definitely have the gene product W, right? So we're going to write them as homozygous dominant for W. They do not have pink, and they're pure breeding, so we're going to say they're homozygous recessive for P. And let's see, do they have the Y and the E gene products? Well, if you see later on in the problem, it says that assume the pink flowered plant is mutant in only one step of the pathway. So we're going to draw them as homozygous dominant for the other 2 alleles. and just assume that it's only mutant in this P gene product. So this is the pink flowered plant. Now let's look at the yellow flower plant. Well, to be stuck at a yellow pigmentation, that means that you're lacking both either the Y enzyme or also the E enzyme. And because either enzyme can bring yellow to orange, that means you must be lacking both, right, in order to be stuck at yellow. So this genotype, we know that they have the W gene product because it can get from white to pink. We know it has the P gene product because it can get from pink to yellow. And then we know that it's homozygous recessive for both Y and E because it can't make either of those enzymes. So we'll draw little y, little e, and that's why those are yellow. So this is the parental generation and their genotypes. Now the F1 plants, what are their genotypes? Both parents are homozygous dominant at W, so the F1 plants are homozygous dominant at W. And then you have little p crossed with big P, so they'll be heterozygous at P. And they'll also be heterozygous at Y and E. Now, when you have at least one dominant allele at all of these genes, that means you are able to make these gene products and go all the way to the orange pigmentation. So let's just look if in the problem it does say that all of the F1 offspring have orange flowers. So our data so far is good. Now it says determine the phenotypic ratios of the F2 offspring if F1 plants are allowed to self-fertilize. So basically what they want is if we're crossing big W, big P, big Y, and big E, little e, and you're crossing that with itself. And that's what self-fertilization is. And what they're asking for is the phenotypic ratios in the F2 offspring. So now let's do the F2. So let's do a simplifying line method. When you're crossing homozygous dominant W with another homozygous big W, the only offspring that the genotypes of the offspring can only be homozygous big W, right? So we're just going to write 1 big W. Now for P, you're crossing big P times big P. Now that's, usually a 1 to 2 to 1 ratio of homozygous dominant to heterozygous to homozygous recessive. But because you only need one dominant allele to get the gene product, and they're just asking us about phenotype, so it really doesn't matter genotype, let's simplify that a little bit. So we can say that 3/4 of the offspring are going to have at least one big P. And that's because a quarter will be big P, and a half will be big P. And then 1/4 is going to be little p. Okay? And that's just, when you just looking at the P gene, that's just a monohybrid cross. Now you're doing, again, a monohybrid cross for Y. So you have 3/4 at least one big Y, 1/4 little y. And then for E, it's another monohybrid cross, right? So you'll have 3/4 at least one big E, 1/4 little e, and then you just do that all the way down. Sorry, this is getting a little messy when there's 4 genes. It's definitely difficult. Okay, now to complete the line method, what you do is multiply across, right? So we're going to do 1 times 3/4 times 3/4 times 3/4. So that is 27 out of 64. And I'm just going to write the phenotype here, where it's going to be big W, big P, big Y, big E. Now remember, that's because if you have at least one dominant allele, phenotypically you're going to make that enzyme. So we'll go through the pathway later to see what the actual phenotype is, like what flower color. But for now, let's just write the, shorthand version of phenotype. So it's 27/64, big W, big P, big Y, big E. Then you have 1 times 3/4 times 3/4 times 1/4. So that's 9/64. Big W, big P, big Y, little e. Now we're going, I don't want to confuse you guys, so now we're going here, right? So that's 1 times 3/4 times 1/4 times 3/4. So again, that's 9/64. And here we have big W, big P, little y, big E. And then 3/4, 1/4, 1/4. So that's 3/64. Big W, big P, little y, little e. Now we're going here. So it's 1 times 1/4 times 3/4 times 3/4. So that is 9/64. And that is big W, little p, big Y, big E. Then 1/4, 3/4, 1/4. 3/64. Big W, little p, big Y, little e. And finally, we're down to our last two here. So you have 1 times 1/4 times 1/4 times 3/4. So that's 3/64. Big W, little p, little y, big E. And then finally, this is the last one, 1 times 1/4, 1/4, 1/4. That's 1/64. Big W, little p, little y, little e. Let me just double check we did all of them. Okay, so now that we have these abbreviated phenotypes, we have to actually go through the biochemical pathway to see what these will be. So let's look back up here. So if you have a dominant W allele, which makes you the gene product W, you can definitely get from white to pink. And as you can see below, all of them have a dominant W allele. So none of them are going to be white. So there'll be 0 white. And therefore, they'll at least all be pink. Now, if you have a dominant P and you make the gene product P, you can get to at least yellow. So let's look for the ones that don't have the gene product P, and those will be the ones that are stuck at pink. So let's see which ones have a recessive P. We have this one here, 9/64, 3/64, another 3/64, and another 1/64. So if you add them together, you get 9 times 3 is 12, times another 3 is 15, times 1 is 16. So you get 16 pink. I'll write that up here. Okay, and that crosses out these 4 genotypic offspring. So these here, we'll just write them, I'll say they're pink. Okay. Now, once you do have that big P allele and you make gene product P, that means you can at least get to yellow pigmentation. Now, in order to be stuck at yellow, that means you have to be recessive for both Y and E. Because remember, if you don't have E but you do have Y, you can still make the orange pigment. And if you don't have Y but you have E, you can also make the orange pigment. So to find out how many yellows that we have, we're going to see which ones don't have either Y or E, or will be homozygous recessive for Y or E. So I'll highlight those in blue. So we're looking for the ones that are recessive in both Y and E, and that is just here. So that's 3/64. So we'll write 3 plants are going to be stuck having yellow pigmentation. And therefore the rest of them are going to be able to make orange pigmentation, right? They have the W gene product to make white to pink. They have the pink gene product to make pink to yellow. And then they have either the Y or the E gene product to make yellow to orange. So these here are going to be orange. So if you do that, it's 27 plus 18. So you get 45 orange. So I'm just going to erase so I can write the answer more neatly. The phenotypes for the F2 generation when you cross two F1 plants is going to be 0 white to 16 pink to 3 yellow to 45 orange. So because there were 4 different genes here and it was a biochemical pathway, it's kind of, there's no possible way to just do this with the dihybrid cross ratios. You just had to do the line method to get these phenotypic ratios of the F2 generation. And then also the P and the F1 plants, I just erased it, but the P genotypes was big W, little p, big Y, big E. And those were that was the pink parent. And the yellow parent was big W, big P, little y, little e. Okay, so those are the answers. Sorry it took a while, but as you can see, once you get more than 2 or 3 genes, that line method does take some time, but that is the only way to do it. It's the simplest way to do it. Obviously, a Punnett square would take forever in this case. So that's how you do this problem, and I hope that this helped. Question 3. The mutagenicity of a new drug is researched using the Ames test. The results obtained are shown below. Which statement is most consistent with these data? So if you look at the data, they used 4 different samples, right? They had distilled water plus liver enzymes, and these are the 2 controls, and then they had drug plus the drug plus liver enzymes. So the 2 controls that included water, you do see a few His plus revertant colonies, but we're going to say that those are just, random mutations. Because water isn't a mutagen, so that's how we know that those are random. So let's look at each statement. For statement A, it says that liver enzymes are mutagenic themselves. Well, as you can see, from distilled water, there was a random mutation rate of about 3 His colonies per plate. And then the distilled water plus the liver enzymes, there was only 4 revertant colonies. So those numbers are basically similar, so we could just say that as well as random mutation, and that liver enzymes are not mutagenic themselves. If they were, then we would see a large increase in revertant colonies from the distilled water to the distilled water plus liver enzymes. So we're going to say that this is false. Statement B says neither the drug nor its conversion products are mutagenic. Well, from the control groups that had 3 or 4 revertant colonies, you see that the experimental groups had 49 and 51 revertant colonies. So, considering everything, that's a pretty large increase in revertants, which means that this drug and its products that from the liver enzymes are mutagenic. So, B is also false because we know that they're both mutagenic. C. The drug itself is not mutagenic, but its conversions are strong mutagens. So if the drug itself wasn't a mutagen, that means that we wouldn't see that many as 49 revertants. So this is false, because even when just given the drug, you see 49 His plus colonies, which is evidence that that is a mutagen. Statement D says the drug itself is mutagenic, but liver enzymes will not increase its mutagenicity. So if you see the drug and the drug plus liver enzyme data are fairly close. You have 49 and 51. So statistically speaking, those are, basically even. So we know, as we said before with the last statement, that the drug is a mutagen, so that's right. and liver enzymes will not increase its mutagenicity, that's also true, because if it did increase its mutagenicity, then in this data here, we should see a larger increase, like maybe 100, 150. So D is true. So we're going to circle D and say that, that statement is very consistent with these data. Now, statement E says both the drug and its conversion products are equally mutagenic. Now, although it only asks for one statement, this statement is also pretty true, right? Because the drug itself caused 49 revertant colonies, and the drug plus the liver enzymes, which give you conversion products, had 51 revertant colonies. So they're basically, both the drug and its conversion products are basically equally mutagenic. So E is also true. So we're going to say that D and E are both consistent with this data. Problem 4. 4 non-complementing bacterial mutants were tested for growth on 4 known precursors in the synthesis of histidine, A through D. Determine the order of the linear biosynthetic pathway defined by the mutant genes. Plus indicates growth on the supplement, and 0 indicates no growth. So I highlighted that these bacterial mutants are non-complementing, so that we know that these are 4 different genes that we're talking about. Okay, and it's asking us to determine the order of the biosynthetic pathway for histidine. So we know that histidine is the end product here, right? Okay, and we also know that it's a linear biosynthetic pathway. So first, let's order the known precursors in this biosynthetic pathway. I know that they're asking us to do it defined by the mutant genes, but let's do it with A through D first. So we know that histidine is the end product here. And if we want to know the pathway from using the A to D precursors, let's look at which has no growth on the supplement. So when given the supplement D, none of these mutants grow on there, right? So we know that D is the first precursor in this pathway. Now, if you look at B, only one of the mutants grows on B, so we know that B is the next in this pathway. Then we're looking for the precursor where 2 mutants can grow, and that's A, which is the next step. And then finally, what's left is C. So this is the order of the biosynthetic pathway for histidine. But now it wants us to define it using the mutants. So let me erase these arrows. And let's look at each mutation. So we know that none of the mutants can grow when given supplement D. But the next step is precursor B, right? Now, which of the mutants can grow on B? If we look, mutant 7 can grow on B. So we know that if mutant 7 was given B, A, or C, that it can go to the end product histidine, which means that we're going to assume that mutation 7 is in this gene product here. So I'm going to draw that as a 7. Now remember, that's because this mutant, if given D, it won't be able to grow because it is mutated in this gene product and therefore can't convert to B. But if it's given anything after its mutation, so B, A, C, or histidine, then it can complete the biosynthetic pathway because it isn't mutated in any of the other gene products. Okay, so now let's look for A. We know that 7 can grow on A, but also mutant 13 can grow on A. And if you look across, it can also grow when given C or when given histidine. So we're going to assume that this arrow here, that's the gene product, is the mutation number 13. Okay? And again, that's because if this mutant doesn't make gene product 13, then even if it's given D or B, it won't be able to grow. If it's given D, it can convert to B, but then it'll be stuck there again. But if it's given anything after its mutation, so A or C, it can complete this pathway. Now let's look for C. We know already that 7 and 13 can grow because it's after their mutation, but 22 is here, can also grow if given C, which means that it must be mutated in this gene product. So we're going to label that 22. And again, if it's given anything past its mutation, so C or histidine, it can grow. But if it's given anything before, D, B, or A, it can't grow, and that's because it gets stuck at that A precursor. And then finally, the mutation that's left is 8, and if you look, 8 can only grow if given histidine. So we know that it must be mutated in this last step, in this last enzyme that converts C to histidine. So we're going to label that as 8. So they want the order of the linear pathway defined by the mutant genes. So our answer here is going to be 7, 13, 22, 8, and then finally histidine. Okay, so it's really important that you know how to read these complementation data and this growth on precursor product charts, because I think a lot of teachers are going to use this to make you determine pathways for biosynthetic. Okay, so that's how you answer this question. Problem 5. 6 strains of mutant yeast auxotrophic for tyrosine are crossed and assayed for their ability to grow on tyrosine-lacking medium. Based on the following data, how many genes were isolated in this experiment? How many alleles are present in the complementation group containing mutant number 5? So let's just draw what these plus and minuses mean. So plus means we saw growth, which means that there was complementation. And a minus means no growth, which means failure to complement. And any pairs that have failed to complement are part of the same complementation group. Okay, so first let's look at mutation 1. Remember that when you're looking down diagonally, you're always going to see no growth, because if you're crossing a mutant 1 with another mutant 1, that means they have the same mutation and they won't be able to complement and grow. So you're always going to see any mutant crossed with itself is going to fail to complement. So let me erase that real quick. So let's look and see if 1 fails to complement with anything else. If you look down or even across in the 1 column and row, you see that 1 only fails to complement with itself. So therefore, it complements with every other mutant, which means that it's alone in its complementation group. So 1 complementation group will be just mutant 1. Now if you look at 2, it fails to complement with itself, which is expected, but it also fails to complement with 4 and with 6. So if you want to double check, you can look in the 4 and 6 columns and see that it's always 2, 4, 6 that are failing to complement with each other. So therefore we know that 2, 4, and 6 are in another complementation group. Now if you look at 3, it fails to complement with itself, as expected, but it also fails to complement with 5. And if you look in column 5, you can see again it's 3 and 5. So 3 and 5 is another complementation group. So remember that each complementation group is actually a different gene. So we have 3 complementation groups, and therefore we have 3 genes isolated in this experiment. And it's asking how many alleles are in the complementation group containing mutant number 5. And we could see here it has 3 and 5, so that is 2 alleles. So the answer here, the first answer is 3, and then the second answer is 2. Now let's run through the true and false section for this chapter. Statement 1. A mutation that changes a guanine to a thymine is known as a transition point mutation. So remember that guanines are purines, and that thymines are pyrimidines. So when you're going from a purine to a pyrimidine, I'm sorry, I spelled that wrong. Or you're going from a pyrimidine to a purine, that's actually known as a transversion point mutation. So this is false, and the correct answer is a transversion. Statement 2. Suppressor mutations are a type of reversion where a second mutation at a different location than the first somehow compensates for the original mutation. So that is true. That is the definition of suppressor mutations, and when they compensate for the original mutation, that means that they give the wild type phenotype. Statement 3. High-energy ionizing radiation can cause mutations through chromosome breaking and formation of thymine dimers. So, high-energy ionizing radiation can cause chromosomal breaking, but this formation of thymine dimers actually makes this statement false. And that's because UV radiation is what causes thymine dimers. So, I'll say causes T dimers. Okay. Statement 4. Inversions prevent a chromosome from crossing over with its homologue, meaning that when inherited, it will not be a recombinant chromosome. So that is true. The next statement, the one gene, one polypeptide theory, allowed scientists to understand the relationship between genotype and phenotype. And this is very true. For statement 6, it says the sequence of nucleotides in DNA correlates in a horizontal fashion with the sequence of amino acids in the protein it encodes. So this is very slight wording, but it actually is false. And that's because it says horizontal fashion. We know that the sequence of nucleotides in DNA correlates in a linear fashion with the sequence of amino acids. Okay? For statement 7, it says, on a lawn of bacteria, plaques are areas of heavy growth. Now this is also false. A lawn of bacteria means that the whole plate is heavy growth, and plaques are actually areas of no growth or lysis by a bacteriophage. So we could say, underline that as the false part, and we could say no growth. Statement 8. While the gene is the unit of function, the nucleotide is the unit of mutation and recombination. Now this is true, it's just, straight from Professor Schmidt's theory. And then for statement 9, it says wild-type T4 phages, bacteriophages, can grow on strains B and K of E. coli. Now this is true. Okay, and that's just another thing, straight statement from the theory video. So if you had any trouble with these true-false, I would go back and look through the theory notes or watch the videos again. But, this is true-false are important so that you know the theory behind everything instead of just knowing problem solving. So I hope that this video helped, and this ends the review for this chapter.